I know, yes. I'll try to go through very quickly. I'll try to keep it to exactly half an hour. Okay, so I am, as you know, because I've introduced myself before, the organizer of the Journal Club, assistant professor of math at Oregon State University, and I'm going to be talking about a more computer science-y technique called min-hash. And before I go on to talk about this, I would first off like to ask you guys, who has heard of min-hash before and or knows how it works? A smattering? Ooh, fantastic. Okay, good. So at least some people won't be lost. No, don't worry, I'm going to go over what min-hash is. What's that? Louder? Excellent. Okay, so first off, we're going to talk about what metagenomic classification is, because that's going to be the main application area for this. However, this improvement that I'm going to be talking about is going to be applicable to many different situations as well. I'll review what min-hash is, the magic sauce that makes all this work, and then applying it to real data. So first off, in case you're not familiar with what metagenomics is, the very basic paradigm is as follows. Use whole genome shotgun sequencing to take some sort of environmental sample, like earth, air, soil, human body site associated sample. You throw it onto one of these fantastic devices, and at an extremely high cartoon level, all the little genomes and different DNA that's floating around in here gets shredded, and all those fragments come off, and you get your sequencing data, which turns out to be tens to thousands of gigabytes in size, as you guys are all familiar with. Okay, so basically, the metagenomic classification problem that I'm trying to solve is as follows. So I have all those reads that now we know where they come from. And usually I'm interested in all microbial organisms inside this environmental sample. And usually people study bacteria, but you also are worried about viruses and fungi and all different microorganisms that could be in your sample. Okay, excellent. You can do many cool things with that. That's not what this talk is about. Talk is about this part. You have all these reads, and you have a reference database of, in this case, let's just say bacterial genomes, just to keep things concrete. And so we have a reference bacterial genome 1, genome 2, et cetera, et cetera, et cetera. And our reference databases are growing at quite the fast rate. So for instance, if you look at some of the more complete bacterial genome databases, we have about 30,000 of them on hand. And the size of these things, that you can think about them as strings, and they can be very short, maybe if you're talking about viruses, like 600 base pair, all the way to maybe 15 megabase pair if you're talking about one of these megaviruses or larger bacteria. Okay, but what we want to do is solve this classification problem and basically come up with just a 0-1 vector on the same basis as this reference database right here, telling us that this bacterial genome 1 is in my sample, this bacterial genome 2 is not in my sample, et cetera, et cetera, et cetera, so you know who is in there. So then you have some idea of, in the environmental sample, what are the main players? What are the bacteria that's in there? Okay, excellent. That's a problem that we want to solve. By the way, if there are any questions at any time, stop me, ask, please. I'm going to abstract this, one, because I'm a mathematician and I like abstracting things, but in any case, it'll make our lives easier if we take a Kmer-based approach. So a Kmer-based approach is as follows. So Kmer is a substring of length k, contiguous base pairs. We're all familiar with this, right? So my set of reads, instead of a set of reads, I'm going to think about a set of Kmers for some fixed Kmer size. And these can be really, really, really huge, especially if you're talking about longer or larger Kmer sizes, like maybe k equals 31 or 51, and you're talking about a soil sample that's 10 terabytes in size, then this set can be rather large. Our reference genomes now, instead of being genomes, are going to be smaller sets of Kmers for the same length k. So again, we're going to have about 30,000 of them. They're now sets of strings of length k. Okay, so I have a set over here, A1, representing genome 1. A set over here, B, which represents my sample. And now, instead of a vector of zeros and ones saying, is this guy in here, is that guy in here, what I'm going to do is use a measure of similarity. So you might be familiar with the Jaccard index. So the Jaccard index basically is a classical measure of telling you how much two sets overlap. So it's the intersection over the union, the cardinality thereof. If it's equal to 1, then those sets are exactly the same. If it's equal to 0, then their intersection, they're disjoint. Okay, fantastic. So let's go ahead and do this in the naive fashion. For each one of these guys, go ahead and calculate this number right here and repeat. Well, each individual comparison that you'd have to do, if we tried to do this naively, you could take this guy right here and represent it as a sorted list, right? Take this guy, represent it as a sorted list, and then just iterate down and see when things match. Well, you got to sort them beforehand, so this is going to be, n log n, where n is whichever one is the largest, which is going to be in this case, which is way ridiculously too slow. Okay, so that's not going to work. We're going to have to be a little bit more clever. So to be able to do that, we can try to use minhash. Okay, fantastic. Minhash is basically, it's an example of one of these techniques called probabilistic data analysis techniques, where, as you probably know in bioinformatics, sometimes you don't need the exact answer, you just need something close. Like you have a threshold and you just want to know, is it above or is it below? So you don't need the exact answer. Minhash is one of these examples. It's an approximation of Jaccard index, but it's super fast. Okay, so it was invented back in the late 90s by Broder, an individual at Alta Vista, way old search engine. He's now at Google. But basically, what we want to do is estimate quickly the cardinality of this green area right here, the intersection of these two sets. So we do this by using hash functions. We're all familiar with hash functions, right? Just a function from a set S to some other, maybe smaller set of integers or something like that. Great, fantastic. I'm going to consider a family of these. So I'm going to index them with a superscript, with an I, and I'm going to consider k of these different hash functions. And then I'm going to use these hash functions to pick out random elements. So randomly sample from A and B. The way that I do that is by using a minimizer. So basically what I do is, okay, well the image of my hash function is numerical, so what I can do is I can find the element that hashes to the smallest guy in here. So like in this case, the thing that hashes to 1 maybe. Okay, so that element is going to be my minimizer, and so I'll put a little min down here, and that's going to be an element of S. I've randomly sampled my set by using my hash function. Now the thing, my clicker has quit working. There we go. Pick smallest element. Key observation here is to note that if you take the smallest element under this hash function of A, and it turns out to be equal to the smallest element of the hash function under B, that happens exactly when that random element that you picked is in the intersection. Precisely when that element is in the intersection. So a hash collision occurs when the element is in the intersection. We'll run through an example real quick. But basically, what you can do is you can use this to then create a random variable that tests for, did the situation happen or did it not? So we define ourselves and indicate a random variable, 0, 1, did my hash functions collide or did they not collide? And then it's pretty straightforward to see that the expected value or average value of this random variable is going to be equal to my Jaccard index. Great, fantastic, except for this random variable is 0 and 1, so it has huge variance, right? So to be able to make this work a little bit better, we're going to do this a bunch of different times for each one of my hash functions in my family. And then sum all those guys up, divide by k, and then that thing is going to be an estimate of my Jaccard index. I realize that I dropped parentheses around this k, so this looks like x to the power k, but it's not. It's just a superscript. Okay, so a real quick example, a real dumb example. Here's a set A, here's a set B, and I have just elements in these sets, A, B, C, D, E, like that. And I'm going to choose a hash function. So here the Jaccard index is actually 1/5. Here's my hash function, A hashes to 1, B to 2, etc. Okay, in A, oh geez, I messed up the color. Oh, I reversed them, blue and red, whatever. Okay, well in any case, A is the thing that hashes to the smallest element. It goes to 1, whereas B and C are hashed to something bigger than 1. B, the thing that hashes to the smallest element is C. These guys are not equal, so our indicator random variable should be a 0. You take another hash function, like this guy, and now, under this second hash function, A goes to 4, B goes to 5, C goes to 1. Ah, that's the smallest guy. That's the element I pick. I go and I do the same thing with B. That hashes to 1, that to 2, that to 3. Ah, so C, oh hey, look, that's the same value right there. So now my indicator random variable should be equal to 1. And now I take this guy, which is a sum of these two, 0 plus 1, 1. There we go. I did this for 2 hash functions, so that means my estimate of the Jaccard index is 1/2. You know, 1/2, 1/5, you know. But in reality, you use more hash functions than just 2. And that will tighten down the variance of this. In fact, you can quantify very precisely the accuracy of this approach. Question?
What is your hash function? Just some random permutation of ordering?
Yes.
Random ordering of that's it?
Right. So if you want to prove results about this, usually you use permutations. In practice, you end up using just a nice family of hash functions.
So hash is just some practical implementation?
Correct. Yes.
Precisely on the previous slide, was for permutations, not for general hash functions.
The which one?
You had the word precisely, like the identity that you, between having the same minimum and belonging to both groups, that's only if the hash function is a permutation or bigger in some sense.
Yes, yes. So for this talk, I'm going to sweep that, the sort of what properties does the hash function need to have to make this work under the rug. But so originally I had permutations up here, but yeah. We actually need them to be k-wise independent to be able to then, you know, make this work as well. So yeah, definitely we have to consider that. Yeah.
So you run several random permutations and then
Precisely.
So you just sample.
Yeah, exactly. This is just a practical way to randomly sample.
Yeah.
Precisely. Yep. And the important thing is that you're using the same sampling on both A and B.
Do you have a bound of the number of samples, some logarithmic number of
Yes.
samples?
Yes. On an upcoming slide. Yeah, go ahead.
If I understand it correctly, so far what you said, everything holds if instead of mean, if we take max. So is there any reason
Yeah.
you are taking mean?
Nope. It's just an arbitrary choice. So basically, it's just pick a random order,
So
take
max will be also.
a specialized element. Yeah, totally. Okay. Example, yada, 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 yada. 1/2, 1/5. So to be able to quantify how good this thing is, you can use your Chernoff bounds. So the Chernoff bounds, the way I'm going to state it is as a bound of relative error. So in this case, come on, there we go. This big expression right here, this is my relative error, right? This was my min hash estimate of Jaccard minus real over real and absolute value, so that's relative error. The probability that your relative error is larger than some pre-described delta, so probability that relative error is large, is less than or equal to some expression right here. And if you think about this, this is e to the minus something, right? So from your guys' perspective, it looks like this. So, for example, if this exponent over here is very large, that means we're going to be way down here in the tail, my probability is going to be small. Probability of deviating from reality is going to be small. If, however, this thing over here is very small, the exponential back up here, the probability is going to be large that you're going to deviate from reality. Yes?
So an alternative approach, which would be probably the first thing I would try, is just have a hash table for all the, basically all the 30K genomes.
Mm-hmm.
They just randomly sample from your new sample and try to see if it's in the hash table.
A hash
Why would that work?
table for all
I
30,000?
mean, yeah, in Egypt, basically what you want to know is you take a random sample from your environmental,
Right.
and then you basically just check if it's there. If it's supposed to be, I don't know, 1 over k, or some epsilon fraction, then you need to do 1 over epsilon times something. Then you can only
So randomly sample the larger set and see if it's in any one of those guys. So you'd have to make a hash table for each one of your entries and then check.
This one you do for any one, for each of them, right?
Uh-huh.
So I'm saying, let's just concentrate on one.
Yes, sure.
Right. So
Yep.
if you concentrate on one genome, you basically just take your large thing and then you basically just pick from it 1 over epsilon times log in order to basically check how, what's the number of what's the overlap that they have.
Yep.
Then you can apply Chernoff bound.
Yes, we're going to do something very similar to that. Yes, and so yes. Yeah, so I'm just rehashing minhash, that Broder introduced earlier, and then we're going to specialize it and then change it up slightly to make it work. Okay, so the cool thing about minhash is, with respect to the size of the sets that you're looking at, it's big O of n to build, and then it's constant time to compare, and big O of 1 for your space as well, constant time.
K, right? Big O K, right?
Well, yeah, so you can, is n changing or is k changing? Yeah, so. Okay, there was a really cool paper called MASH last year that looked at applying this, doing a nice implementation, bioinformatic key implementation, and you can do fun things with it, like take all NCBI genomes ever and cluster them by using this approach, and then on one single CPU, so 32 CPU hours, and I think it was like 20 gigs of RAM or something, so something very small. You can get these cool clusters where you can't read this because it's way too small, but these are species names, and you can get, hey, look, you can cluster almost perfectly, have that clustering agree with species if you do this. So, hey, fantastic. But my main application area is metagenomics. I want to apply this to metagenomics, and it's not so good. Why is that the case? Because if you remember the size of those sets that I was talking about earlier, so the sample is huge, right? Hundreds of billions to trillions of elements in it, and then the individual genomes are smaller. So it turns out that your Jacquard index is always going to be ridiculously small, right? Like 10 to the negative fifth or smaller. And if we were doing that exponential curve thing, turn-off bounds are then going to be useless, right? Okay, so we're going to have to be a little bit more clever. If we take a really sloppy relative error right here, and we want to be 95.5% sure that we don't exceed this relative error, we're going to need more than 10 to the 7th hashes, which then it slows down. It's not so good, way too many hash functions. Because the compare part, as you're saying with a k, in terms of k, is k log k. So can we do better? So this is your idea, basically. So can we focus in on this part right here, and instead of looking at sampling this whole union, let's just look at A intersect B and A right here, and randomly sample from this part. So it may not be precisely your idea, but similar. Okay, so the cool thing is if we do this, putting aside practical implementation, because we'll get to that, we've reduced the set that we're randomly sampling from super large to not so big. Doing this is using containment instead of Jacquard index. So basically, how much of A is in B instead of how much do A and B overlap, which is a lot larger. So as we said with the turn-off bound, it's this little term right here, the 10 to the negative fifth, that's messing everything up. So if we could replace that with this containment, that would be fantastic, and we'd get it. But can we do this efficiently? So if we took our indicator random variable, and instead of asking do hash functions collide, you can now say, let's take this randomly selected element and test, is it in B? Is it not in B? And you could do this. You could do it super naively, like, okay, you could do it in kn time, right? Or you could hash up all of B. Oh, but shoot, B has like 10 trillion elements in it. So you want to make a hash table off of this? Oh, okay. Well, we can get it to work. So let's ignore those sort of practical things for a minute, and let's just see if my turn-off bounds will make sense. So if I do this, does my minimizer, is it in B or is it not in B? My expected value is the containment like I want it, and I get the same expected values, and I get the same probability bound, and hey, look at this. I was able to replace my Jacquard index with containment. We're in business, except how do we check this when B is so large, ridiculously large? So that means Bloom filters to the rescue.
You gain nothing from taking the minimum here. It's just a random element from B.
Yeah, randomly select an element. Yeah, it doesn't matter. So whenever I write this, I just mean select a random element. Yep, these are random variables, so I just have to select them. Yep. We're all familiar with Bloom filters, right? This I'm going to somewhat assume. Just a quick recap. These were invented by Burton Bloom back in the 1970s, given some set S. Bloom filter, which I'm going to denote S with this little tilde, is a probabilistic data structure, which is usually used to reduce space. that has the following properties. You have no false negatives. So if you have an original set, if you have an element in there, it's definitely going to be in your Bloom filter. But you have some small probability of false positives. So you have some element that wasn't originally in your set, but turns out to be in your Bloom filter. Cool thing is that you can characterize this false positive rate quite easily. So the probability that you have a false positive is some expression involving parameters that are used to build this Bloom filter right here, and you can specify that and you can control it. What I'm going to talk about subsequently works for many other set membership test probabilistic data structures. However, I'm using Bloom filters here just because they're so ridiculously easy to analyze and so easy to control the false positive rate and everything like that. So usually you use this for significant space savings. What I'm going to do is I'm going to use it here for a constant time set membership test. If you have a more favorite one, just insert that here. So putting it all together, you take random elements of A and you test, is it in this Bloom filter or is it not in this Bloom filter? Great. That works. The only thing is we have to be very careful about how we apply the turnoff bounds. We have to carefully go through the calculation to be able to incorporate this false positive rate to be able to see how that affects the turnoff bounds. But first, a question.
So, some clarification. So, the
Yes.
Bloom filter comes to eliminate the hashing, or what exactly it eliminates or replaces what?
Right. So right here, this is not my set B, right? This is a Bloom filter of my set B. So it has two advantages here. One is it allows constant time membership tests. So is it in or is it not in? And the second thing is it also reduces the size. Yep, so it's like a really nice hash table, like you were saying.
Replaces what? B, the original B?
Yes.
Bloom filter.
So B is my original set. B tilde is a Bloom filter of that, right? Which is smaller in size and quicker to check if an element is in it or not.
In the price of some fraction, of course.
Right. Some false positive rate P. Exactly. Yes?
In the previous slide, notations, what was small n and capital N?
So you use multiple hash functions to construct this thing. M is the number that you use. N is the number of bits that you use because it turns out this is just a bit array. So it's the number of bits you use. Yeah. Okay. So you very carefully go through Chernoff bound stuff, and this is going to be your proper estimate of containment index now using this approach. And we get this expression for the right-hand side of my probability of deviating from reality. So if you compare this, first off, this term right here, this is basically 1, right? P is small, that's my false positive rate. C over C plus P is basically 1. And then this right here, hey, that's something nice and big. A lot bigger than my Jacquard right here. How much better? Well, you can go through and you can, give these some name T, that's what your desired confidence bound is, and actually go and recalculate the number of hash functions required by this approach and the classic min hash approach. You get some expressions. The whole punchline is that, so when I say we, this was mostly by my PhD student back at OSU. He's done fantastic stuff with this, but basically in realistic parameters, in realistic settings right here, you get the following. So let's do a plot where on my x-axis right here is my relative error bound that I don't want to exceed. And then on the y-axis here is going to be the number of hash functions. So these expressions over here for the classical min-hash approach and using this containment approach right here, and you get the following. So for these different parameter values right here, notice this is a log scale. And this blue here is the containment approach. This is the classic min-hash approach. So down here for lower values, instead of needing 10 to the 4th hash functions, you need 10. So it's a pretty cool, significant improvement of reducing the number of hash functions that you use. However, you might say here, you're comparing apples to oranges, right? Because this approach is estimating containment index, whereas that other approach was estimating Jacquard index. Well, then all you have to do is all Human has to do is do some very other careful calculations and figure out how you can estimate Jacquard from containment in an efficient way by using this other thing called hyperlog, which I don't have time to get into. But basically what you do is you just tweak your relative error from delta to delta prime in some nice way, and you end up with another expression that, once again, basically a 1 here, a large value right there. And look at this. We're estimating Jacquard index, and our bound is based off the containment. Yes, there we go. So now I can do an exact apples-to-apples comparison, and we get plots like the following. On the x-axis is my relative error. On my y-axis is the probability of deviation, so basically this part over here. From the Chernoff bounds, this blue and green line, which are completely overlapped, are both the Jacquard estimate from this approach and the containment estimate from this approach. The red is the classic min hash. The way that you read this is that if you want a 20% relative error, then there's a 0% chance that you're going to get worse than that. If you were to use classic min hash, you would have about a 90% chance of exceeding that. And same thing depending on if the size of your relative overlaps of sets is different, yada, yada, yada. 5 minutes left, so we can zoom through space-time complexity and then talk about application a little bit. Cool thing is even though we're using a Bloom filter, you save a whole lot of space with this because you can calculate the number of bits that are required for the classic min hash approach, and it's based off of the number of hash functions as well as the number of reference genomes that you have. For our approach, you pay some penalty off of the Bloom filter, and then you have the same term right here based off of number of hash functions that ours use, which is exponentially smaller. And it turns out, since G is so large, then using these various formulas right here, you actually get a, what is that, 10, 100, well, you know, it's 0.06 times the space. So it's a heck of a lot smaller space in all. And of course, to do it in time, you know, it's k log k, where k is the number of hash functions. We use a whole lot less, so it's a whole lot faster. Great. So what can you do with this? The cool thing is you can convince yourself that these Chernoff bound calculations are actually accurate. So we can take a bunch of random strings. So I take one string and I copy it, and then I append on a random string over here, and then let the size of that, you know, change so I can range over a bunch of different Jacquard values. And then I look at the classic min hash approach right here, comparison between true and estimated, doing the containment estimate of Jacquard right here, and hey, look at that, woo, better. Let's go ahead and see if this is actually going to work in our simulated situation right here. So let's simulate a metagenomic sample, and then let's go ahead and just randomly take one of the genomes that we simulated here and check if we can see what its Jacquard index is. Also do it brute force so we have a ground truth. And if you do this and you simulate yourself a very small metagenomic sample, then this is what you get. So this is a plot of, this is number of hash functions that you use versus your mean relative error. And we already know that the more hash functions that you use, the better the approach gets right here. These error bars and these plots are the means right here because I did this many, many times over. Blue over here is the containment approach that I'm presenting. This red is the classic min hash. And you can see they're basically indistinguishable. However, once you start to get to realistically sized metagenomic samples, you know, 1 million, that's not even realistically sized, that's still ridiculously small. 1 million reads, the second the bigger set grows bigger, you get much less error. So you get this huge, massive drop in the containment approach. So this is sort of demonstrating to you that if your sets are relatively the same size, just use classic, no need to like construct Bloom filters on the side and everything. If, however, the sets you're comparing are very different sizes, then yeah, you probably want to be using this containment approach. To see if this is actually going to work, I took a very small subset of the soil metagenome, and I wanted to see if I could pull out different really low abundance organisms. So I was like, viruses, let's look at viruses in this soil sample right here. So I took about 5,000 viral genomes, and I ran this containment approach using just 500 hashes with, this is my false positive rate for my Bloom filter. And I said, okay, let's just look at the one with the biggest containment. So I picked out my virus, the biggest containment, and it turned out to be this leaf curl disease virus. And I'm like, hey, that actually sounds a little bit reasonable, right? Because Iowa Prairie soil metagenome, leaf curl disease virus, and looked up in some paper, and yeah, this is like endemic to the area. So that was great, but is it actually in my sample? So first off, using all those different formulas from beforehand, I can be more than 80% sure that I'm not off by some ridiculously large factor, which is great. If I wanted to have the same confidence with the classic approach, I would need about 10 to the 8th hashes. Ooh, question?
What happens when you have errors?
Yeah, so this is a real sample, so it's going to have errors in it, definitely. I guess what happens when you have errors? I need to quantify that a little bit. What happens to what when you have errors?
People don't care about sequence errors.
Yeah. These viruses and everything evolve so quickly. I can't tell the difference between errors and actual mutations. So, no.
To the model, you could ask what happens if you have.
Yeah. So you could make that thing sloppy, for instance. So instead of saying is it in the is it in the set, you could say is it within one edit distance of it and things like that. Sure. Yeah, you could do that. I haven't looked at it though.
So another question, so your
Yeah.
largest containment index is about 2.5%.
Yeah.
The aids that you found only matches in 2.5% of this virus, do you think?
Well, when subsampling to only 500 hashes, this sketch of that genome was covered, about 3% of it was covered. But the thing is,
You took only the 2.5 million reads, no?
yeah, I only took 2.5 million reads. So it's a very small subset and a very small, genomes right here.
Actually, what's your K for the Kmer?
31. Yep.
So errors is a big problem. Because sequence errors, you don't expect to have that many.
Yeah, errors when only 31, yeah. Yep. But in any case, let's go ahead and use sequence alignment to see if it's actually present. So let's go ahead and, this is one of the reasons why I took so few reads, is so that I could do brute force sequence alignment on this thing. And I did find 288 reads from this genome sitting in there. And you could do one of these coverage plots right here. So you have the reference genome on the outside, and this is a little windowed box plot of the number of reads that have aligned to it. And, I don't know, this looks at least somewhat encouraging that perhaps this virus is actually present in the sample. What I'd have to do is probably go back and actually use the full one to see if I can get full coverage.
So how much coverage do you get when you do this? Of the, or how much?
Oh, the updated version gives me the mean. I can't remember what the mean coverage is off of here.
Yeah, just trying to see, I don't know what the comparable measure would be for the containment index when you think about.
Right, so if your coverage here is, like a perfect 1x, then your containment will be 1. If your containment is like a perfect, so each base pair is exactly, covered half the time, then your containment would be half. Yeah, contiguously. Okay, 2 minutes over time. So containment min hash, appropriate for estimating Jacquard when one of the sets is a lot bigger than the other. And you definitely want to use some sort of probabilistic set membership approach because it's more accurate, use less space, and it's faster. So why not? I have an implementation of it up. If you're familiar with Titus Brown's Kmer project, I'm working with one of his postdocs to get it implemented into that. And the cool thing is we've been working on making the paper for this completely reproducible. Oops. So the cool thing is if you go into any of the scripts and change any of the parameters, you hit make paper and it'll go and it'll pull those parameters and actually put it in the paper. The conclusions might, not be accurate anymore, but all your plots get updated, everything gets updated. So I thought that was kind of cool. So, but in any case, yeah, thanks for your attention. Now I am standing between you guys and coffee, so perhaps coffee? Sounds good. You can ask me offline.